Hi, Fred Bright, USDA ARS in Raleigh, North Carolina. My question is, you mentioned in one of the preamble speeches that it's a 7-day time period for getting data for whole genome data, and that's longer than the 4-day period for PF pulse field gel analysis. What are the stumbling blocks? Why does it take 7 days when you can do the sequencing rather quickly? What are the main stumbling blocks, and are there potential solutions to speed up the process?
The process is longer. There are more steps that you need to go through. You need to isolate the DNA, you need to do the library preparations, and then you need to do the actual sequencing. And you all need to fit it into a normal workday workflow. So it just takes the time that it takes, but I'm pretty sure that as the sequencing technology evolves, the turnaround time will definitely also go down.
Yep, I would agree with that I think at the moment, as we're introducing it and using it, it's still some stages a little bit clunky, but there's lots of rooms for improvement. So in my lab, the isolate comes, and then on a slope, then we culture it, and then we grow it, and then we do the DNA prep. And I think we'll get to the stage where you can do the DNA extraction straight from the slope, and the sequencing methods will get much faster, the library preps and everything. And I'm really confident that it will speed up the turnaround times that we're getting at the moment. Because I'm very impressed with the analysis times. Once the sequence comes off, when it's analyzed, it's really very fast, a matter of like minutes or hours once you've got that raw data going through the pipelines.
Can everyone hear in the back? I just want to make sure this microphone's turned up enough. It needs to, we'll try a different microphone.
Other questions?
Yeah, sure.
Yeah. Good morning, Betsy Barine, OFW Law. Thank you for your presentations. One of the things as I think about the mystery of genome sequencing and how we have the clinical samples and then we have research, and I'm very intrigued by a lot of the retrospective cases that we're seeing and the analysis of what we're learning about outbreaks. I'd be interested in your opinion. I see genome sequencing from a research tool of, let's take Hurricane Harvey, change the environment of a certain area. Can we use this technology to better understand how organisms in these environments may affect farms, produce, and different types of stress? Do we see that we can use this tool for research? And you all have a lot of experience, but to expand this in ways for us to better understand how these pathogens are living in an environment and then living in an environment of stress. And how do we, if we're food companies, better understand our incoming load and those stresses when we have a food safety system that are designed to set at a certain base? I'd just be curious in your thoughts.
Okay, so I think there's some opportunity in that. So I'm going to rephrase that. You have a food safety system, it has a certain heat kill step, and you monitor your incoming material. Now, whole genome sequencing theoretically should tell you, oops guys, our supply chain changed and now we have a much more heat-resistant salmonella because it has a heat-resistant islet. So you're going to have to change your heat treatment or your critical control point because of that, and you do that real-time monitoring. I think that's where metagenomics, if we fast forward 8 to 10 years, is going to come in, that we can adjust shelf life labeling based on the incoming load of spore formers to the grow at low temperatures, we need we need to adjust our critical control points potentially, and ultimately we're going to move that into predictive. We're not just going to test and say we do, because there's still that time lag. And we say, when you have this conditions, there's a high likelihood of X, Y, and Z. So that sort of routine monitoring will happen, and it is already sort of happening. I think we're finding some of these strains and really starting to think about, do we need to adjust our CCPs or our interventions in some of these conditions.
I think, Ruth, did you have a question in the back?
Good morning. It's Ruth Petran with Ecolab. Martin, and maybe Peter can weigh in, but I thought it was interesting, your idea of thinking about doing epidemiology different. And I think there's certainly some sense in that, but, so thinking provocatively perhaps, so what would that look like? Or what could that look like, given that we've kind of done it the same way for forever?
Man, that's why I call it a mystery, because I don't actually have a simple answer. Because it really, the issue is if you have two cases and you had a massive recall that's already in the newspaper and you ask them, did you eat product X that, by the way, was mentioned in the newspaper on the front page yesterday, those two people probably will remember that, even if they didn't eat it, maybe. And then what do you do? Then is there an outbreak linked to those, or maybe there's another source? It's very hard. So with these small numbers, it really partially is going to be different types of statistics. There are better statistics to deal with small numbers. What we're completely lacking is a way to sort of integrate. We have different statistics for describing SNP differences or allelic differences, and odds ratios are what we use to classically interpret epidemiological data. We need to develop some statistics that take those two data sets together and interpret them together. I think some of the future will relieve of some of those issues as we have better records. As we go to e-commerce, people will have perfect receipts, perfect traceability, and that's going to fix some of that potentially going forward. But in the short term, I think there's going to be considerable uncertainty around it. But I'm looking forward to see what Peter and others say.
Is this working? Can you hear me?
No.
No.
You need to use that one.
Okay, can you hear me now? Great. In my experience, that we tend not to get the recalls and then we find the cases. And what we are finding is that because of the preciseness with the whole genome sequence analysis, that it's able to rule cases in and out of your investigation with such preciseness that it makes the analytical epidemiology much more powerful. Because previously, if using things like pulse field gel electrophoresis, we would have included cases that were not part of the outbreak. And so all of their food histories that we would have got would have been included and would have sort of confound the investigation. And that really is not happening anymore. So I think it has advantages for epidemiology in being able to do that. But I totally, I completely agree that it is just a tool, but it's proving to be an extremely powerful tool. And across Europe, we're able to identify cases which are common parts of outbreaks. Like we have a Salmonella enteritidis egg outbreak that's involved over 4,000 cases in multiple countries across Europe, with the potential to prevent those cases going forward. And without the sequencing, we never would have been able to have got that information. So whilst we're also identifying smaller outbreaks and outbreaks over a longer time, we're also able to identify much larger outbreaks with the ability to prevent those going forward.
I totally agree with what has been said, but I also want to remind you that there are different epidemiological methods that we can use for different situations. Of course, we're going to use the case population studies comparisons more frequently than we have been. Probably it has always been a screening tool, and it will remain a screening tool. But we will still also be using case control studies. We'll do case comparisons that are the routine for Listeria investigations, and we will also do cohort studies. Increasingly, when we're having big outbreaks, we are looking for clusters within the outbreaks so that we can do a powerful cohort study instead of doing a real case control study. And we're going to refine all the tools we have. But I also want to remind you there's one more kind of types of outbreaks that we are increasingly investigating, and that is the ones that Errol mentioned, the retrospective outbreaks, or the ones that are driven by finding an isolate in a product and then matching it with clinical isolates. Even though some of these clinical cases may be several months or even years behind, if we know exactly what product it is we are looking for, it's easier to do the epidemiological follow-ups, and that has also helped the investigations a lot. And then I will also just add a caveat also, whole genome sequencing is not the solution to all our problems. Some outbreaks we would not be able to detect with whole genome sequencing. In the United States, we recently investigated a large salmonella outbreak associated with a herbal supplement, kratom, an opioid, or opioid agonist, and there were 6 serotypes involved. We detected the cluster because one of the serotypes were pretty clonal, and we could detect it by whole genome sequencing and by PFG. But there were 2 serotypes, Salmonella Thompson and Salmonella, or very rare serotype, let's say that. But those 2 serotypes, by whole genome sequencing, they were extremely diverse, with more than 50 SNP differences, or up to 50 SNP differences between the strains that were involved in the outbreaks. In the case of the Thompson outbreak, or Thompson part, we could have detected it by PFG. The rare serotype, we could only have detected that by looking at the serotype. So whole genome sequencing, there are instances we do not know much about the production and distribution of this product. So there are situations where whole genome sequencing doesn't give us all the answers.
I'd just like to touch on one point Peter made. I think one area where, at least as a bioinformaticist, I've seen WGS make a big impact is, in the retrospective realm, but now at WGS, just improvements to food questionnaires, so asking people what they ate. We have leads on to ask if, cheese from this specific creamery, produce from this specific source. We may not catch it this time, but we have much better food questionnaires for the next time we see this pattern.
So, and as we know, as the industry, we tend to then get really nervous when we talk about matching a food product to a clinical isolate, because where do you draw the line knowing, is that the only food product that had that sequence in it? I mean, how do you know that it's Cargill's starch versus somebody else's starch? Do you go and look at all the starches then? Because I think when you're doing epidemiology, it's not going down to the level of where did all the ingredients come from now in everything that they ate. So this is why us as in the industry get very nervous when we hear this, because granted you might fix one facility, but that doesn't necessarily take away and help public health if you have multiple facilities causing this problem.
Okay. Right, I think that's a really good point. And, but I actually think that sequencing will help you identify the source and where the source came from. So if it's, so that you're not just putting all the emphasis on one production facility. If it's, for example, we've got a Listeria outbreak ongoing at the moment to do with frozen sweet corn, and we have identified one particular very large company that distributes it. But actually, we think that it's coming in from the corn itself, which is not just going to that one particular company. So it's enabling us to widen that, and it's a combination of both the food traceability and the sequencing results that will really help us get to the bottom of the problem and to put in preventative measures that are much more effective. And then once you've put those measures in, I find that sequencing is really helping to say, has this gone away? You can, because when you do get a positive with a patient after the interventions have gone on, you know exactly where that came from, where, what the problem was, and you can go back and check it, because that's what we're seeing in our large outbreaks.
So then I just want to pick up on that because, of course, a couple of weeks ago I learned about there was an outbreak in Belgium with school children from the lunches, and they knew it was the plant that made the school lunches for 11,000 kids that sickened 500 kids. But I don't know as of today, but at that time, they had no idea what the source was, where it came from at all. And so I struggle with that as to how good is whole genome sequencing. And I do recognize we did say it wasn't perfect yet. And yet, I think, is the key word. I'm not saying it won't get there. I also struggle with how long it took us to find the source of the lettuce outbreak here in the United States. And I keep hearing how strong whole genome sequencing is, but then you hear and you see these things happening, and then you start questioning, well, is it really as strong as we thought it was? Why is that happening? So I recognize to see one case matched with maybe a product, but I'm not seeing this silver bullet yet with whole genome sequencing.
Well, whole genome sequencing isn't the silver bullet. I completely agree, and you need to do your epidemiological follow-up no matter what. And even epi can be very complicated, and it's not perfect either. So that's just the way it is. Just a comment also about how it is that we are using these matches between a food isolate and clinical isolates. Often, if it's a recent food isolate, we will also ask specifically about that product with patients that when we're interviewing the patients. But particularly if it's a food product that has been an old food product that's in the database, we are not so much focusing on the actual producer, but more on the food commodity, and make sure that when we are doing our epidemiological follow-ups, that we include questions about that particular food commodity, and not so much on the specific producer.
So can I, just as we're on this topic, just ask quickly, Pam and I work in industry. If we get those unfortunate phone call one day from CDC and they said, we think we've linked your product to illness with whole genome sequencing, what questions should we be asking?
Show me the data.
Perfect.
Yeah, why? Yeah, you need to ask us, why do we think so? And I think if we call you from CDC, at least, we are going to tell you why we think you are the source. And then, of course, you need to question us about why, if you don't understand the arguments that we're presenting to you, you need to ask us, make us explain what it is we're doing.
In Public Health England, we've had two major companies come to us with sequences from pathogens they've isolated and ask if we've seen this type before or had any cases. And actually, on both occasions, we haven't seen any at all. We had no matches when we did our Snip analysis. So it can be used, and it is being used for reassurance as well. Sure.
Just, I think you've probably heard from the FDA, CDC, and others, but if that call is happening, it's probably not just genomic data. We've given in multiple presentations just that it's the genomics plus the epidemiology plus the traceback. So there are probably multiple lines of evidence that would support that finding if that call is happening.
as I would sort of step by step, I would split it into three things. Number one, what isolates do you have? What human isolates, where did they came from? What food isolates, where did they came from? Intact package, not what type of package? What environmental isolates, where do you have them from, etc. Then number two, what subtyping methods did you use? I would actually, honestly, we have so much standardization, the SNP difference, the allele differences, I'll believe in most. Those are pretty reproducible. 20% of my questions go to isolates, 70% go to epi, 10% go to SNP. I think we focus way too much on the uncertainty around SNP and whole genome sequencing when we really need to focus on the back end, the data interpretation when we ask those questions. So then ask a little bit about, how did you match it, how many SNP allelic differences, a little bit on that, and then, okay, so how did you draw the conclusion? Was it what sort of study was it? Did it link to humans in addition to sequence data? What other sources did you investigate? Do you understand the full supply chain? Where could it have come in? If it's found in a finished product in an intact package, it's one thing. But even then, the questions become a little bit as, could it have been in other intact packages and types of the product? And then I think it's really important for industry to help CDC and FDA understand the complexity of the food supply chain. And I think that's my, and I think regulators in the US and public health agencies have gotten better to listen to industry, but you guys need to listen to industry and the supply chains. Those supply chains are extremely complex. Every time I look at one, I get surprised. And they change. Just because you knew supply chain for product X 2 years ago doesn't mean you know it today. Just because you knew consumption patterns from 10 years ago, they changed. Industry know how they change. It needs to become that two-way road. So I think from industry side, I would then want to try to really get them to really understand the supply chain, to find other potential sources.
Yeah, that was very helpful.
The population survey is being repeated as we speak, so we're going to have some more recent data very soon. I'd say we use the industry also when we are doing the tracebacks, because the industry knows the distribution much better than we do. So we do talk to the industry actually, even before we implicate anyone. And that is really the industry's opportunity to put us into the direction where it will become much easier, clear, who is the culprit here. Is it you that we're talking to, or is it somebody else? And most often it's probably somebody else.
We'll go back to the audience. I think we have someone been patiently waiting.
Yes, can you talk about the implications of culture-independent diagnostic tests?
So far, we haven't had big implications of culture-independent diagnostic tests, but it's definitely a threat. The culture-independent diagnostic test is basically PCR panels that are used to diagnose foodborne infections in humans. Increasingly, they are very fast and they are pretty accurate, but they will only give you identification to the pathotype or the species level. And theoretically, we don't get any isolates, and then we need the isolates in order to do the whole genome sequencing and do the cluster analysis and identify the clusters. But so far, at least in this country, we've been able to establish a system where the culture-independent diagnostic positive specimens are being cultured, and then we get the specimen. So what it means is that there is a delay before we get the isolates, but we do get most of the isolates, and we will be able to also use it for cluster detection. But we do know also that this is not going to continue forever. At some point, if all the diagnostics in this country moves to CIDT, as we call it, there are nobody who has the resources to do all the culturing follow-up. So we have to develop some methods that by themselves will also give us all the subtyping information and strain characterization without a culture, and that is some kinds of metagenomics. We are working on it, FDA is working on it, academia is working on it. There are different approaches. I'm not going into any details, but at least enough to say there are some technological breakthroughs that needs to happen before this is going to be really efficient. The sequencing technology needs to be cheaper, faster, more efficient, and the biotechnology definitely also needs to improve. I don't know if anybody have any other.
Yeah, at Public Health England, we are looking at culture-independent methods, but it is at an early stage, but I think that there's a huge potential there. But obviously there's lots of problems when you're looking particularly for foodborne pathogens when it may be, in the minority, and trying to get the data out, you might be able to detect certain genes but not link them to the one particular pathogen. So there are lots of challenges, but I think going forwards with metagenomic methods is going to be a really exciting, interesting area, or it is a really exciting, interesting area, but there are quite a few challenges at the moment to overcome.
Peter mentioned the metagenomic research programs. I think one sort of interim solution is, at least for the foreseeable future, we're going to continue to get the food and environmental isolates, and certainly from a bioinformatics perspective, comparison of human metagenomic, across hypotheses about, single isolates from food or environmental sources. That's a much more tractable bioinformatic problem than metagenomic to metagenomic comparisons. And so there is some light, I would say in some sense, as long as we keep getting the food and environmental isolates in the database.
Do we have one on this side of the room? There's a microphone. Okay.
Edith Wilkin, Loprino Foods. My question is a little different twist. This is an international body, and I'd like to think we don't live on an island. Here in the U.S., certainly there is the dynamic going on where FDA is asking industry to contribute to the public databases. There's a lot of expertise in industry, and there's quite a bit of work that is being done for our own purposes using this tool. But there obviously is a concern with the metadata that would be attached to any information that industry is contributing to the databases. I am curious if anybody on the panel has any idea or can inform us what's happening in other parts of the world specific to this dynamic of industry contributing to public databases.
I can speak for Cargill being a multinational company. Cargill is not contributing any data to that database. And I think in outside of the world as well, I think this tool is not as strong probably between the EU and North America. I'm not sure how much South America is using it, nor Africa, Southeast Asia. We don't hear about it as much in those regions where we are.
I would just like, I can't comment on the international perspective, but I can say from FDA, certainly we are trying to provide opportunities for industry to analyze their data, that if they generate it, should they want to compare it to the public databases. We would hope that through, parties outside the FDA, those comparisons, organizations, nonprofits could support that kind of activity.
I think step one's got to be not data sharing, but the ability of industry to use and access the data. Right now, we're in this tug of war, right? I mean, industries have some data. They say, you show us your data first to CDC and FDA, not just the limited metadata. And regulators and public health say, you give us your data first. And everyone's like, and we're not going anywhere. So we got to start somewhere differently. And one got to start, and I would encourage the regulators and the public health agencies put out data with more metadata that make it more useful. That's not trivial because there is, confidential information. But I think a company that has an environmental isolate can search a database saying we got 2 cases, close enough match within our distribution area, we're going to shut down that plant. They want to have that ability. Right now, they really don't, because there isn't enough metadata there to understand that in most cases, and the data get out too slowly. So CDC, get the data faster in the public domain where people can use them and do exactly that case study. That's the answer in my view.
Okay. Public Health England makes its data publicly available, and it's doing that at the moment as soon as it's generated. So it's going out very quickly. And but I do think it's a case in Europe that industry isn't putting its data into the public database. But I think at the moment, I'm just glad that they are using sequencing, that industry's increasingly using it, because then it makes it easier when matches do come up, or we can look at it, that we're able to quickly analyze that data because the sequencing's been done.
I don't know, Martin, CDC not uploading our data in real time. I think we are more or less, there might be a few delays every now and then, but for most purposes, I think all data from CDC and from FDA and from USDA is uploaded to NCBI in real time. But that is definitely not the case all around the world. Yes, Public Health England is also doing it, but many European countries in public health, they cannot upload in real time. And the European Food Safety Authority is definitely not uploading much in real time. But that doesn't mean that they don't share the data with each other or with us. There are some non-public databases, and we are also building some new ones that we can access, and we can do some comparisons. But of course, the ultimate goal is to get everything out in public domain so everybody can analyze the data in real time. Our experience from United States has been only positive. I can't think of any negative effects that we have had of sharing our clinical or food safety or food regulatory data in real time. It works, and yeah, we should do it. And I think it is going to happen at some point, but it is a matter of building trust and making sure that, or people need to realize that it's not dangerous to share the data.
So, I mean, it's the sequencing data part of it, right? It's the metadata is the other part we need. So do the data come out, you sequence a human assay, put it in a public space, do you attach at least the state and the date of the case to it at that point? Or is that coming later? Because that's the thing, without the metadata, it's not very useful.
You don't get state information from PulseNet. You can't do that because it's almost PII. But you get the region and the country of the United States where the isolates are from. And what was the other thing you were asking? Date. You get an upload date. So if it's a recent upload date, it's probably pretty recent.
I think from an FDA discussion, certainly on the minimum metadata, we had years of conversations through Global Microbial Identifier, including government, academics, and industry, to come up with a minimal metadata set that everybody was comfortable with. And so certainly that's what we share at the FDA. We're working to make that richer. There's certainly, I don't think industry has ever asked us to put a company name or an address on there, so that hasn't happened. But we're trying to put information, for example, type of production facility, zones, to make the information more useful to industry, but it takes time for us to pull that from our other databases and put that information in the public domain.
Yeah, next question.
Oh, yeah, hi. So Robin Atkinson-Dunn, I'm with the Utah Department of Health, so welcome to Salt Lake City. I have a question. There's an entire field of microbial evolution, and I'm wondering how that's being applied to whole genome sequence data and trying to get at some of the concerns that were voiced earlier. Martin's smiling.
So, how to say? It's basically being dumbed down for use. We're not considering mutations aren't mutations, they're different. They're synonymous and non-synonymous. There's sites that are under positive selection. We're ignoring all of that. We're trying at a very superficial level to remove recombination, which can give you a lot of SNP differences very quickly. The different pipelines differ slightly with how they do it. That is one of the areas that can give us sometimes problems, how we address that. So the information is used but we're not doing the formal sort of evolutionary analysis, particularly to interpret SNP differences. And I do believe there's some more information out there in the SNP data once we dig in deeper, rather than just saying A is different from B by one nucleotide. We need to know, we can use it where it is, what it means, etc. But that's really very much on the fine-tuning part, and that's where it's used. You have a cluster, you have, and then at the very back end, for less than 1% of cases, you might go down to that level in terms of using actual true phylogenetics to understand the data. No, because you have the generation time issue. The big issue is the generation time issue. You can have, I give you a hypothetical salmonella in a dry plant. It's going to double 20 times a year. Same salmonella in a poultry farm, active infection, it's going to double every half hour, 50 times a day, times 365 days. Within a year, you can have a range of 1,000-fold difference in terms of generation times. That gives you 1,000 times different rates on a per year in terms of SNP accumulation. How do we get at that? That's the big one.
Just following on that, certainly sort of the first use of the database is really for surveillance and hypothesis generation, and it works very well, for that. I'll also just make the point, we're making all the data available, so our hope is that different research groups can take a deeper deeper dive into the data and start to help us, understand the microbial evolution we're seeing in the data, and that's why we're putting the data out there.
Yeah, I would agree with that I think those kind of studies looking over time and what's happening and the evolution are being done, and to understand that evolution in different conditions to better inform what is going on.
Where's our next question?
Hi. Hi, thank you. Lucia Analich, South Africa. I'd just like to make a comment rather than a question, if I may, on the previous question, which was about the data sharing outside the United States and the EU. In Africa, it doesn't exist, basically. But in South Africa, considering the listeriosis outbreak we are currently busy with, there has been whole genome sequencing occurring from a public health perspective, and that's how we managed to find the outbreak strain. Since then, of course, industry is also now doing whole genome sequencing, and there are many more laboratories coming up with the actual technology in a private capacity. However, there is absolutely no trust between industry and government, because the way this outbreak was handled and communicated to industry was dreadful. Government was not prepared to share any data with the industry and instituted a recall without any information sharing from their side to the private sector. So they have asked the private sector for information prior to finding the source, but the private sector was not willing to share any information that they may have had. So yes, we don't do that sharing. I'm hoping that we will learn out of this outbreak that we can actually start to work together a lot better as industry and government together. I think it's going to take some time, but I'm sincerely hoping that that will be one of the positive things that will come out of a dreadful situation where this morning I had the most updated numbers. We're now at 214 deaths. So this is critical, and we do need to have better collaboration between industry and government to solve this problem and any future ones coming up. Thank you.
Hi, Julie Hendigas. So we're hearing a lot about outbreaks and scary words like implication and recalls. Don't you think it's beneficial to know the genetic makeup of what's in your facilities so that you can know about biocide resistance, you can know about heat tolerance? And if you think about what the hospitals are now doing with their surveillance program, looking at antimicrobial resistance and possibility of transmission rates, I would think it would be good that the industry starts looking at that kind of stuff. And then you can almost do it internally before your product goes out. So if you see that you have a quat resistance, you can take care of it and do it right away. And that's where whole genome sequencing is beneficial.
We're not there yet with whole genome sequencing. First of all, I mean, one of the first questions was, why does it still take 7 days? So there is no such thing of adjusting then to fix your quad or your temperature, your critical control points in today's world of whole genome sequencing. It takes way too long, and the other problem is it's very expensive. So again, we have designed a food safety system in this world based on HACCP, based on preventive management systems. And this is a tool to verify that those systems are working properly. So it would, yes, totally agree with you. We would love to be able to put a stick or something, and in 30 seconds it spits out that this one is more resistant to quat and we need to up, the concentration or change the heat. But we're just not there yet on that. And so, again, someday, we hope. And also, to my understanding, whole genome sequencing is not there where we're going to say, is this sequence now more virulent, less virulent, affected by heat more? We're not getting those kind of data feedback yet with what we're doing.
Well, just to add to that, so I see in the future what could be created are PCR panels, but what we need are those sequences to be able to find genes that could be targeted. So in your facility, instead of having to sequence everything, you could run a PCR and say, okay, I have a quat gene or I have an acid gene. So then it's not the cost of sequencing, it's just the cost of PCR.
Well, I mean, we do, whatever testing we do, we are running most of it is molecular that we do. I'm giving a talk in the next session in this room about what methods are used, but if you've never worked in a facility, it's a whole different world, and we have very many different stresses coming at us. And unfortunately, today's tools, as well as whole genome sequencing, none of them are perfect. None of them are real time. So we would love, and we are looking forward to the advancements that whole genome sequencing is going to be making, just like everything else. But we have to balance the time it takes to get results and to keep our products moving. Cargill makes a ton of bulk products. We make over 1.5 million pounds of starch at one facility every day. I mean, this is not, some cases of lipstick that we can store in the warehouse for a week while we're testing, and it's not a big deal. There's a lot of food being made out there, and it's got to be moving, and we can't wait for these results. So 25 years ago, we asked for real-time methods. We're still asking for that.
I think you're drawing a picture of the future that we're getting there. But I think another of the mysteries back to the title is, how do we sample properly? Because you're talking about 1,000,001 pounds of storage, and you're saying we should sequence everything, totally agreed. But how do you sequence everything from 1.5 million pounds of storage produced every day? It's really non-trivial. So the future there is of an incorporation of sequence data with then improved data analytics to do prediction. The sequencing, as much as we can, is an intermediary stage. And I think a lot of the investment that I see in the food industry now is building the data analytics capabilities, that when these tools become cheaper, because I think there's also a disconnect. There's a disconnect between me as an academic and Pete and others saying, it's $50 to sequence an isolate, it's not expensive. To me, getting one isolate and someone wanting it turned around in 2 days when I need to run my sequencing run for $2,000 because I can do $50, but only if I run on 96 at the same time. So there are all these sort of discrepancies, and they add up. So I think industry is moving there to building the infrastructure, but what I see is it's really very much on the data analytics part, and then some on the sequencing part. And not saying Cargill is doing sequencing, but I think more companies doing sequencing than they say.
I agree with Pam that you probably can use whole genome sequencing for process control for products that are going out now, but you might be able to use it to solve some inherent problems that you see in a facility. Persistence of a strain. Why does it persist? Well, that's Cargill. That's good. That's good to know. Buy more Cargill. But yeah, problems like that, if you don't understand why your control measures don't work, then it might be a good idea to sequence it to figure out if the sequence could explain what's going on here.
Hi, this is Bala Ganeshan from Mars. I represent the Mars Global Food Safety Consortium. I work with them as a senior scientist, and also the Mars IBM Consortium for sequencing the food supply chain. I would like to add a couple of points of clarification. I think we are really excited by the quality of questions that have been asked around in this forum so far. So in terms of addressing some of these futuristic questions regarding whether what these SNP differences mean and where we can take them, just to put a little bit of a futuristic bent on them, that just taking the SNP differences to classify the strains or identify the differences is one thing, but knowing where that SNP is and what, whether if it is a heat resistance gene or a heavy metal resistance gene, whether it's conferring more superiority or inferiority to the strain based on where the mutation is and where the survival is. The point is that we can actually do that today. We can start taking this type of data and take a peek into the future and at least gain a little bit of perspective. Of course, we have to, I don't deny that we have to follow it up with the functional studies and pilot studies and all of that. But from an analytics standpoint, I think we just need a little bit more perspective on the SNP differences being also biologically important beyond classification and identification. The second point I would like to mention regarding some of the diversity-related questions is that with the Mars IBM consortium, there has been this very nice 100K pathogen sequencing project where 100,000 foodborne pathogens have been sequenced, and more than 25,000 salmonella or close to 13,000 Campylobacter genomes have been sequenced, and some of the analyses has been displayed as graphs in different forums in the last 2 years that I'm aware of already. We already know that looking at these SNP differences in evolution, as someone had asked earlier, it's not necessarily this nice and beautiful bell-shaped curve. It's more of a ragged curve across at what rate, which region where SNPs are accumulating, and there are questions still running around what serovar or what species mean with genus. We don't think there's a question about it, but what the species and subspecies and serovar definitions are from the phenotype versus genotype. I think we are in a position to answer them, but also to be able to understand and appreciate that alongside this craggly bell curve, there is also some leading or following smaller bell curves or smaller bars of more wider diversity than has been previously appreciated. We don't have a presentation doing here in this meeting, but I would just like to, if anyone wants to read the papers, I will be happy to point the references to them. Thank you.
Any additional questions?
Hi, Justin Filardo from the University of British Columbia. I have a question about pipelines, and I'm wondering how much pipeline affects the final results, and can you compare between pipelines, and how does one choose what is the best pipeline to use for as a researcher?
Yeah, we do that a lot. That's what we do for a living. I mean, so certainly we have bake-off comparisons between the respective SNP pipelines. Should we mask, regions where there's a high density of SNPs? Does that correspond to phages? Does that correspond to repetitive elements recombination? We spend a lot of time thinking about that. So from our groups, I would say certainly from the FDA, CDC, and NCBI, it doesn't really matter which pipeline you use. There may be a difference in the 5 to 10% range of the SNPs that are detected, but that's not at a level, 9 versus 10, we're not really going to come to a different conclusion from a genomics perspective, at least, about the relationships of those isolates. So we spend a lot of time on those comparisons and those bake-offs. As far as what's optimal, I think that's actually where you have to choose a compass and go with it. So if you're going to look at sort of thresholds or relationships and start to put that information together, that's where it is critical to pick, be consistent about your assemblers, be consistent about your sequencing technology, and be consistent about your pipeline, because if you're not, a lot of the things you're going to see may be artifacts. You certainly wouldn't want to run an MLST with 2 different types of annotation. All the differences you're going to see are artifacts of that and not, so don't just take the data from GenBank. And run your analysis. You may have to redo a lot of the work in-house to get the answer.
So what SNP pipeline should you use as a researcher? It depends, of course, on the question you're trying to answer. Right? So if it's a public health question, you want to just reproduce basically the conclusion that CDC or FDA comes up, you of course will use their pipelines with their sort of settings. For some research questions, you might use different pipelines because you're interested in recombination events, like big chunks of DNA coming in. You don't want to map them out because that's where your interesting stories are as a researcher. So I think that the goal, you need to define your goal first. I think more important, if you take the existing SNP pipeline, so the CDC or the FDA give you very similar results, unless you use the wrong reference genome. That's actually the bigger concern. So in most of these pipelines, you get SNPs, but you need to have sort of a template to call those SNPs. We call a reference genome. If I'm using some Listeria serotype 1,2A and I'm going to use a 4B genome as a reference genome, I'm trying to put a puzzle of the Alps together with a picture of the Rocky Mountains. Ain't working, okay? So your reference genome becomes really important, and what we see is that's where a lot more of the big discrepancies come in, is with choice of reference genomes.
I'll just say on Martin's point, one project we do have at the FDA, we are looking at if you're familiar with the pathogen detection portal, we're looking at all the large SNP clusters and try to at least do a PacBio closed genome for each of the large SNP clusters for Salmonella and Listeria and put that data out there so that if people want to use our pipelines and pick a reference, it may be easier because there will be complete genomic references for the different SNP clusters.
I might add also, for public health purposes, where we are doing a whole lot of analysis, speed is of essence. And we don't have time to find the right reference all the time, at least not in PulseNet. So we're using a database instead and using the MLST approach.
Hi, MT from Biomehue, Latin America. I just want to complement the question she has raised about what is being done in other countries. And I can just say to you guys that in Latin America, we may have few industries that do whole genome sequencing, very few, that usually data is not shared, and that in government levels, we have very, very, very few labs that are able to do it. So for our reality, it does not exist, not in regular basis, just to complete the information.
But if you export to United States, whatever isolates that are in there and are identified by FDA or USDA, they will end up in, the sequences will end up in NCBI in 5 seconds.
It's a little longer than 5 seconds, but we certainly try and share the data in real time for all the import testing, as Peter mentioned. So.
Yeah, maybe, but it's not informed and public and anything.
That's correct.
Behzad from iFish Chicago. I have a question about the high throughput sequencing, and it was mentioned it's a tool, and I was wondering if it's just a tool. There is nothing else, differentiating it from other tools that we've had before. And also, what is your advice for industry in the meantime that we still are dealing with the mysteries surrounding the technology interpretations and problems, the challenges like being expensive or interpretation is hard. So what is a different company what different companies can do in the meantime, in the short term, in the long term. Should they invest in the technology? Should they learn about it? And I appreciate your input.
I'll just comment first on the tool question. It is just a tool, but I think one thing that is unique about it is the data is standardized and it's much easier to compare. For example, if you've tried to standardize mass spec data across different instrument manufacturer platforms, it's a complete nightmare. But with sequencing, we can largely do that. And so we can share data across agencies, and it's been a game changer on that side. As far as the advice question, I may kick that over to Martin there.
I think industry needs to know the tools and needs to know how they can access them quickly when they need. And the extreme examples, there's an outbreak linked to you, or FDA, USDA finds 3 isolates with the same subtype over a year, your plant gets shut down, you need to do a root cause analysis and prove where this isolate came from. And in many cases, that means you have to show by whole genome sequencing that you found the source of that isolate. So what industry should do is, you're used to mock recalls. Do a mock recall, a mock outbreak with whole genome sequencing. If you have some old isolates that you've banked, which some of you may have not, make them part of the mock recall. Figure out if you need to access whole genome sequencing, how we get there. Who are we going to send it to? How quickly they're going to get its results back? Are we going to send it to some university that's going to lose it and it's going to take them 4 years to get back to us? Is there a commercial supplier that can do it quicker? Are they not doing the analysis good? Run through a complete mock recall with access to whole genome sequencing. So you know who your experts are, you know who your labs are, they can do that and they can help you when it gets there. At a minimum, you can invest in your own tools and your own MySeq and whatever, and your own pipeline, your own bioinformatics people. And I have a feeling that's happening to us. We do offer commercial whole genome sequencing, and we get some isolates for whole genome sequencing. I have no idea why we're getting them. We never hear again, and I think it's sort of a test run. Hey, we know that these 4 are the same, we know this one is different, see whether you're getting the right answer. They're probably getting sent to 2 or 3 different labs, trying to figure out how quickly people get results back so you are ready when that other shoe drops. That to me would be the most important advice.
Yeah, I mean, like we said today, it is very expensive to set up your own lab as an industry. And you need to then hire people who are experts, like bioinformatics. This is not a simple, the industries just can jump into making this part of our routine labs or our pathogen labs. This is a huge investment. So today, typically, we use the experts that are outside, and good idea to know and do all that. But really, the bottom line is still today, whether we use whole genome sequencing as that tool, the best thing is to make sure we understand completely the methods we're using and to make sure they're fit for purpose, especially with the sample types we're testing. I don't think people put enough time researching the methods they're using to find these problems and get better. They're just doing the same thing they've done for 20 years, and we have to get away from that. We really need to have a better understanding of what's going on in our plants, in our products, and with our testing.
To answer the first question again, whole genome sequencing is just another method, of course, but it's such a powerful method. You get so much more information than just the subtyping. Basically what we can do is replace many of the traditional molecular and phenotypic testing methods by this, and that is what makes sequencing cost efficient in public health. And also, related to the second question, I say because it is so powerful, I hope that the industry, especially the industry that is not really embracing whole genome sequencing yet, will think about whole genome sequencing if they have a problem with microbes in some way that you might help solve by characterizing the strain. Then ask yourself, will sequencing help? And if you can't answer it, ask somebody who knows about it and who can help you. And there are always somebody who can help you.
So maybe we have some time for one very last question.
Thank you. Larry Cohen, Treehouse Foods. Following up on Martin's point and Pam's piece, from an industry perspective, I think there's a large number of people out there that have would agree that doing like a mock recall and understanding where you would send your sample, et cetera, would be a good step and a good verification tool. I'm past that point, and I think others that are with me were to the point where, like you said, where you're receiving some isolates, you're testing them, then you never hear back from them again. So I'm at that point. If I get a positive with a pathogen, a product pathogen, or I have a zone 1 that might be a persistent strain, I have the whole genome sequencing done. It's expensive, et cetera. I just have a few strains that I might be dealing with. What do I do? How do I treat it differently than my PCR strain identification, where I'm going for the root cause and trying to identify how to get it out of the plant, or it might be coming from an ingredient, et cetera. So I'm now at that what to do, because I've sent it to an expert. They've told me the importance of it. Now what?
So in many cases, you're really not getting initially more information out there than you're getting out of a PFG in the old days. It basically tells you same, different, then something else. Then you have to start your root cause analysis, figure out why you have the same 2 times. Now, with whole genome sequencing, sometimes then what you should do is do further analytics. Say, okay, is there something unique about that strain that might explain why we have certain issues that, with a pattern with PFGE you can't say. You want to be careful with that. I mean, we heard a lot about sanitizer resistance. Be careful with this. There's a difference between a research definition of resistance and a practical definition of resistance. I get very excited if a strain goes from 0.5 ppm resistance to 2 ppm resistance, because now it's resistance against quad. Just you guys use 400 ppm or 200 ppm. So it's really practically irrelevant in most cases. So just keep that in mind. Now, I'm not saying it's in all cases. It can be predictable, but you got to be careful also in terms of the interpretation. They aren't always as straightforward as you think. So you want to try to sort of get additional information out of it. But I think for industry, in some cases, many cases, PFGE will answer 95, 98% of the questions. You don't always need whole genome sequencing. And if that is accessible to you, cheaper, faster, stick with it, at least for the time being.
It's one thing that is distinct about the whole genome sequencing from PFGE is just, once again, the public databases are accessible. So if you're dealing with a particularly difficult problem, you want to look at attribution, source tracking. The public data is out there for you to interrogate it and perhaps come to those conclusions or hypotheses too. Yeah.
And I guess that's the other thing. You can ask whoever did the sequencing for you, or you can ask me saying, can you search it against the databases, tell us whether there's some matches. But then, the paranoid people out there, well, as soon as you log into a database, is Big Brother at FDA going to know Larry from Treehouse logged in and searched the whole Now, I'm going to put it, everyone thinks that, right? I'm just the only one who says it. So that's why you come to someone in academia or so, and no one knows it was Larry from Treehouse who searched the Salmonella database, and it's like, what's going on at Treehouse? So there are some ways we get around. So that's probably what you want to do, and get some information. How you then going to have that communicated back to you in the practical reality of running a business becomes another one. And I'm going to be, again, practical. You probably want me to call your lawyer to call you. You all know how it goes, but some people here might not. Okay. Yes. But I'm just doing it for everyone here because not everyone is at that stage. That's how it really goes.
It's all the same database.
Well, I just wanted to say thank you for everyone for participating. This was a really great session. And please join me in thanking our panelists one more time.